Hi everyone, I'm Aurelien Pelisiser from IBM Research on ETH Zurich, and I will present cell type specific gene regulation in rheumatoid arthritis. So nowadays there have been a huge increase in the dataset of RNA sequences, and this already has led to the discovery of many novel therapeutics in the context of clinical settings. And for that, the main method are gene-wide association studies, for example, or differential gene expression analysis. And also recently a lot of work in gene regulatory network inference. But all these methods, they have a major drawbacks is that they disregard information about the cell types. And the problem is that usually these datasets, they come from a bulk dataset which contains several cell types. So it's really hard to extract this information from this data. And the regulatory mechanism across cell type might differ widely. So here is what we will present today. We will explain how we can estimate the cellular composition in synovial tissues of rheumatoid arthritis. Then we will explain how we identify key drivers driving rheumatoid arthritis in a gene regulatory network. And the last part will focus on the inference of these networks, cell type specific also. So to do the work, I didn't make any new measurement. I used the public data. And the first one is a big data set of around 200 synovial biopsies, which is very big for array data set, and it's good because we don't have to do a batch correction or batch normalization, which removes some biases. And the second one is AMP phase 1, which is an accelerated medicine partnership, which provide actually very interesting data because this is single cell data, but they also have bulk data on cell type specific. So I will come back to that later to explain what is important. So the first part, how do we extract this gene expression profile from synovial tissue? Actually, there is many ways to do that, but there is a tool called Excel where it was pre-trained on 64 cell types, and it's testing some enrichment score for each cell type to see if in each sample it's a specific cell type is enriched or not. And once we do it on a synovial tissue, which is like the first dataset I presented, we can already see some huge differences here. For some cell type, you have, for example, AKT cell, natural killer cell. There is very few in rheumatoid arthritis samples, but quite like around 10% in control. So control, we use like some healthy patient, also some patient with osteoarthritis, which is like a completely different kind of arthritis, which is not autoimmune disease. And in this list we could discuss for a while. Like for example, there is all these immune cells that are known already to be important in RA, like T cells and B cells, but also you have like this kind of cell like eosinophils or parasites that this one have not really been described in literature. So the problem with so this really shows that between RA and non-RA sample, you're gonna have a huge difference in the gene expression in the cellular composition. So how do we identify key driver gene in RA in such a context? So we will combine like differential gene expression analysis with prior knowledge from literature, from different databases, and also some public network already published in literature from different methods. So you have like the human interactome, like which compute like the protein interactions. We also have some large scale study like RIMBANET or GIANT, which are Bayesian neural network. So how the key driver works in our context? So it's very straightforward. So we start from two things. We start from a list of associated genes to array, and we use a gene regulatory network, and then we test in this network that for each gene, we test that if a gene has a causal relationship to many associated genes, then we consider it as a potential key driver. So this is very simple. And what we do is that first we need to build this gene associated list. So to do that, we did two things. First, we took like differentially expressed genes from literature. We combined 6 meta studies plus our own study, and we took the overlap. If a gene is in at least 2 lists, we put it in the list. And the other thing we did is that we checked all these databases that have information about RA. So for example, the drug bank for the gene target, or like the disease or dysgenet. And again, if a gene is at least 2 databases, we take it as a list. We take it in the list from literature. So we have 2 lists that we'll show later. So here, one list with around 100 genes, the other one with 250. And then we have to use a network. So for the network, we use the giant network because giant network has a specificity of building a cell type specific network. So they build a cell type network for like dendritic cell, T cell, B cells, and they also have a tissue specific network. So we took all of these and we run the key driver analysis for all of that, which for that we use the merge-omic library. And what we obtain is this. So here's this plot. We can take some time to talk about it because it's quite interesting. So first, the original list, which is like the 2 lists I mentioned, the literature and the differentially expressed gene list, they have a very low overlap. So this is the red thing you can see here. Both means like there is no overlap between the 2 lists. But after we run the key driver analysis, we end up with a very high overlap. So for example, in dendritic cell and NKT cell, we have more than 70% overlap between the 2 KD analysis, even though the initial list was very different. So this tells us that the key driver analysis seems reliable. And depending on the cell type, you had some cell type where there were much more key driver found than others. For example, for dendritic cell, we had around 2,000 key driver genes, but only 500 for eosinophils. And the same for the tissue. Some tissue have more key drivers than others. And so this already gives us, like for each cell type, it gives us a list of key driver genes. And yeah, so now let's talk about a completely independent method that we can also use. So it's good to have independent methods so we can combine these two together to get the overlap list and then really have the best potential key drivers in the end. So for the second method, what we do is that we build our own network, and for that we use Panda, which is a method that is building a biparty graph where basically a transcriptomic factor is linked to several of its target genes through directed edges. And this network is built as follows. So you combine 3 sources of dataset. The first one is a prior knowledge about transcriptomic factors interactions. And the second one is a list of the motif target genes for each TF. So this is prior knowledge that we get from database. And then from that, you add the gene expression matrix from the RA synovial tissue. You combine all of them and you try to maximize the agreement, which is method described in this publication, and you end up in with your network. And another interesting thing that we do is we use something called Lioness, which is making sample-specific networks. So, for example, in this example on the right, I have 5 samples, and 3 of them are RA, 2 of them are control, and you can make a network for each of these samples. To do that, the method is simply using a leaving out method. So you compute the network with combining all of the sample, and then you remove one sample and you combine the network again, and you check the difference. And this gives you information about the weight of that sample specific network. So again, you can check the publication to see the details, but this is a very powerful method because now that we have a list of gene expression and a list of edge weight for each sample, we can run some differential analysis between the two groups, like the array and the control group, both for the expression and the edges. And once you have that, you can check for the correlation between, you can check if there is any correlation. So if a TF has a high correlation, there is a high correlation between his edge weight and the gene expression, we can expect that this TF is important in the context of rheumatoid arthritis regulation. So it look like we can make a ranking of the TF, and you can see here. So yeah, it's quite blurry, but you really have a positive correlation between the differential expression and the differential edge weight. So we did that for the entire synovial tissue, but you can also do that for each cell type. So in the AMP phase 1 dataset, you have data about B cell, T cell, monocyte, and fibroblast, and so we made a network for each of them. And interestingly, what the point is supports the point that I explained in the introduction is that each cell type has a very different regulatory mechanism. So here, for example, the differential gene expression is very different across cell type, and you have the same thing. The TF regulatory score across cell type is also very different, very low overlap. So this tells us really that we should really consider each cell type independently. And so what I gain at the end is a list, for example, of promising transcriptomic factors, combining the two methods above, so combining the key driver analysis and the Panda network analysis. And interestingly, for example, in the top 15 of this list, there is like nearly half of them that are already known to be involved in literature. Yeah. So this tells us that our method is quite reliable, that the fact that we find half of them that are already known is a good point, right? And then it means we could explore like the other ones that are in orange here that are not that described in literature. They could be interesting targets for potential treatments. And another thing I can show here is that the correlation between the two methods I proposed. So the key driver analysis score from the merge omics analysis and the Panda score from the gene regulatory network analysis, we can see that if a gene has a very low, if a TF has a low key driver score around at 0, then it typically have a much lower score than if it was a key driver for the Panda analysis. So this also confirmed our two methods that are consistent with each other. And finally, another very important thing is when you want to develop a new treatment, you want to have some genes that are specific to some cell type. You don't want your treatment to affect all the cell type at once. So what you can do, you can check the gene expression for each cell type for this specific gene. And if you find the gene, for example, this one, FOSL1, that is expressed mainly in fibroblasts, but not really expressed in other cell type, this could be a very good candidate for your treatment. So as a conclusion here, we provided cell type specific regulatory information, and we combined different approaches to increase the reliability of our approach. And at the end, we obtained a list of potential cell type that can be very useful for experimentalists who wants to try new treatments. And actually, our collaborator, Persio S. Gulko, who is a medical doctor, already tried some of the genes in this list, and we had some very promising results. Also, I would like to thank, of course, both of them for the support through this project. So Maria Rodriguez Martinez at IBM Research is my supervisor, and Persio S. Gulko is a medical doctor doing the experiment on the experimental validation of my work at Mount Sinai in New York. So now we are planning to test a lot of these in vitro to validate my approach and find new, and this could be used in the clinical setting with in vivo experiment, maybe in mice or in humans. And here I want to mention that we focused mainly on fibroblasts, but this approach could be really used for any cell type and also other disease, not only rheumatoid arthritis. So there is a lot of potential to be discovered, and yeah, we are looking forward to this in vitro validation. So really thank you for your attention, and if you have any questions, don't hesitate to reach me on my email through the ISMB app. So goodbye everyone, thank you very much.